This will be the second part of the nervous system. We'll continue on. We'll look at the action potential, and then we will look at the chemical signaling that takes place at the axon terminals. So we saw previously, when we saw the action potential, once it's triggered, it is non-decremental, so it won't lose steam as we go down the axon here. Okay, and we saw we get a rapid depolarization and a rapid repolarization with this little bit of hyperpolarization. Right, so we'll look at the intricacies of what makes this take place, the timing, the players involved, and all that to make it so we can get an action potential sent down the axon. One thing about the action potential is it is a all or none phenomenon. Okay, and this is kind of like the light switch analogy. At the axon terminal, or sorry, not the axon terminal, at the axon HELOC, right, that initial stretch where all those voltage gated sodium channels are, all right, it is either we are having an action potential or there is no action potential. It is all or none. Okay, and it will go maximally, meaning it will go all the way or not at all. All right, if we just reach negative 55 millivolts, we just reach threshold, the action potential will look like this. If we've got a huge stimulus and we reach negative 40 millivolts, which is much greater, guess what? The action potential will still look like this amplitude. It will not be a larger amplitude. So once we reach threshold, boom, the action potential is on. It's going maximally. So that's all we got to do is we have to reach threshold. We'll see how this gets coded a little bit later. Okay, but in terms of the action potential, either switched on or not at all. Okay, they're gonna, if we measure down the axon too, it will always look as this say. We're not going to lose the amplitude of it. Okay, so no matter what, once we reach negative 55, okay, we can reach positive 30 and it'll still have the same amplitude down the axon. Okay, so all or none. Excitability of the neuron, rapidly we're going to be, as you can see, we're going to have a rapid depolarization and a rapid repolarization, and we'll see when we look at the action potential there is a hyperpolarization phase. Okay, but from this slide here, light switch, either the light is turned on maximally or not at all. It's not a dimmer switch. I already discussed a little bit of the players that are going to cause this action potential, okay, but we have voltage gated sodium channels. Okay, we already talked a little bit about this at the trigger zone, those voltage gated sodium channels. Okay, what's going to be Q to open up these voltage gated sodium channels? Negative 55 millivolts. Threshold. What's going to be the Q? So we have a second voltage gated potassium channels. What's their Q to open? Negative 55 millivolts. Threshold. Okay, and so these are the two main players to give us that big rapid depolarization and then rapid repolarization, and we have this little hyperpolarization phase also. Okay, but the two main players are going to be the sodium channel and the voltage gated potassium channel. Okay, you can see there's a little bit of difference of these, and this is going to come into play to creating this action potential. Okay, but if we look at the action potential, something similar to this, and you see we have three main phases. We have a rising phase, that's going to be our depolarization, And if you remember, what did we say? Which ion's going to be involved in that depolarization? We're going more positive, so sodium rushing in would cause a more positive environment. And what would cause the falling phase, the repolarization happening here, okay, and after the hyperpolarization phase, that's coming here. If this was resting, you can see we're going below resting here. Okay, but what's going to bring our membrane potential more negative or bring it back down? Yeah. If we have potassium ions rushing out, bringing with them the positive charge. Okay, so we got these two players. Now we're going to look at how these two players result in this action potential. All right, before we look at the whole voltage changes, the membrane potential changes taking place during the action potential, let's look at the players in this. And we got our first is the voltage gated sodium. You remember we're going to have voltage gated sodium channels, voltage gated potassium channels, and what is their Q? They are gated, and the Q is that threshold value, 55 millivolts. Okay. And what happens? Let's look at the sodium channel. The sodium channel looks a little bit different than the voltage gated potassium channel because it has this activation gate and it has this inactivation gate, this H gate. Okay. And so you can see in this state it is closed, but it is capable of opening because once this activation gate opens up, then sodium can rush in. Okay, so we hit negative 55 millivolts, that activation gate opens up, and there is sodium, lots of sodium outside, very little sodium inside, and sodium can rush in. What you got to realize is there are many high density of these channels on the membrane down the axon, so you can have lots of sodium. You have lots of pathways in which sodium can rush in. Okay. So sodium can rush in, and this is very rapid. This opening of this activation gate is very quick. Okay. This inactivation gate is a slower. It still is triggered by negative 55, but the most rapid thing to happen in this is this activation gate to open up, allow sodium in, and then ultimately this slower inactivation gate, okay, will come in. By slower, we mean by like a millisecond. All right, we're talking milliseconds, not here this opens, 3 seconds later this comes in. This is within milliseconds. So it's just boom, open, boom, inactivation gate. Okay, because they are both triggered by negative 55 millivolts. Okay, no matter what, the trigger is going to be negative 55 millivolts. We have to have the same trigger for this timing to all work. But what the difference is this is very rapid to open, this is slow to come in and plug up the hole, right? Because once that inactivation gate is in, sodium cannot come in. The other key thing is for this channel to be able to open again, we have to reset it. This inactivation gate has to come down and we have to reset to look like this again. Okay, and so that takes a little while for it to reset. So it can't be bam bam. We can't have a right, millisecond after we can't open this channel again. We have to reset this channel before we can open up the channel again. Okay, this is going to come important when we start talking about the refractory periods here in a bit. Okay, but just realize very fast activation gate to open, the slower inactivation gate then comes up, plugs the hole, and when we're in this state we can't open the channel. We have to reset it, which takes a little bit of time to get back into this state. Now looking at the voltage gated potassium channel, you can see we don't have that inactivation gate. All we have is basically a gate to open up, and potassium, because it's high inside, can rush out. What's key about this channel? Its Q to open is negative 55 millivolts. Okay, its Q is negative 55 millivolts. But just like that inactivation gate, the voltage gated channel is slower than the sodium channel to open. It is slower. Okay, so once we hit negative 55 millivolts, the first thing to happen is the sodium channel is open. All right, that activation gate pops open, sodium can rush in. And then right around the time the inactivation gate, that slow inactivation gate is coming into play, we're going to have the slower potassium channels on the membrane opening up. All right, and then the potassium can rush out. The other thing is as they're slower to open, they are also slower to close, so they have a delayed closing. Okay, so they're delayed to open, and they are also somewhat slow to close. Okay, so keep that in mind when we start looking at the membrane potentials, or the changes in membrane potentials when we look at the action potential. All right, so here we go. Let's put this all together. If we were to measure in an axon, if we were to measure one spot and looking at changes, membrane potentials taking place during an action potential, this is what you would find. All right, we start off at our resting membrane potential. Okay, we get up to threshold, and remember, threshold is where, what is threshold? Threshold is that negative 55 millivolts. That's when you're going to get opening of those voltage gated channels. Okay, and if you remember, the first thing to happen, the most rapid thing to happen, is those very rapid voltage gated sodium channels open first. So the first thing you see happening is this rising phase. Right? It's becoming more positive. And how do we become more positive? If we open sodium channels, sodium rushes in with its positive charge, bringing the membrane potential up. Okay, bringing it closer, the inside closer to the outside. So we bring it more positive. So that is the rapid opening sodium channels. So we have, this is P is for permeability to sodium. When those voltage gated sodium channels open up, the permeability to sodium increases greatly, and we can allow sodium to rush up. What happens in those sodium channels, they rapidly open, and then that inactivation gate slowly comes into play and will stop the sodium influx. And right around the same time that sodium, those slow inactivation gates to plug up the pore at ball and chain, the slower potassium channels are starting to open. If potassium, the permeability of potassium becomes greater, potassium rushes out, taking with it its positive charge, so we start heading back, repolarizing in the negative direction. Okay, so potassium channel, slow to open, but now it's open. Okay, and we're coming down here. Remember, the potassium channels are slow to close. So we rush right past resting membrane potential because we're allowing all this potassium, and we get a hyperpolarization phase. Okay, then as those slow potassium channels start closing, all right, that sodium potassium pump is going to be pumping out. We have that leak channels, and we get back to, there's no new action potential, we get back to resting membrane potential. Okay, so again, the first phase, those voltage gated sodium channels are rapid to open. Boom. that slower inactivation gate comes into play, just as the slow voltage gated potassium channels come in, and now potassium's rushing out. All right, and they're slow to close, and once they get closed up. The other thing to watch out for is here is where that inactivation gates come into play. And if you remember, sodium, those voltage gated sodium channels, when they are in that inactivation gates in play, they can't reopen. All right, so there's a period of time, all right, from here to say here, there is a period of time in which the sodium channels that would be at this position here are inactive. Okay, when they are inactive, so when this potassium's coming down, the sodium channels cannot reopen, right? Then they get reset. When they are reset, we could have another action potential. Okay, so keep that in mind when we start looking at the refractory period. So this one just kind of gives you another look of what's happening. Here the dotted line is the change in membrane potential, but you can see what's happening with the sodium. We have sodium rushing in, that's causing that rising phase, and then you can see sodium permeability. This is the conductance. Sodium's moving in, it gets stopped. All right, there's very little moving in. Just as sodium's getting stopped, we start getting potassium moving out. So it is those ion movements that are causing these changes in the membrane potential, and the ion movements are controlled by the opening and closing of those voltage gated channels. Again, just looking at ion basis, sodium conductance, sodium moving in, okay, is high during the rising phase, and then potassium moving out is high during the falling phase. So again, it is the ion movements that are changing the membrane potential. First it's sodium, then it's potassium. Okay, sodium's causing the rise, potassium's causing the fall of the repolarization. So what we're seeing here is looking at the voltage gated sodium channels. What we're talking about is, say this is the axon here. And I have a voltage gated sodium channel here. There's a little inactivation gate. And say this is where the cell body is over here. Or this is the axon HELOC. And then I have another voltage gated sodium channel here. Okay, this is almost like a positive feedback loop of opening these channels. If you remember, if we took a probe here and took a probe down here, the action potential is non-decremental. Remember, it doesn't lose steam. It is the same. And that is because we're re-upping the signal along the way. This axon has high densities of sodium channels all the way down the axon, has high density of voltage gated channels, or voltage gated potassium channels all the way down. Okay, but remember, so how do we keep this going in one direction, right? We don't want the action potential to come down this way and then come back towards the cell body. We want it heading from the cell body down to the axon terminals. Okay, we want the signal going in one direction. Okay, and how is that achieved? Remember that inactivation gate. Remember that inactivation gate, once it's in place, the sodium channel is inactive until it resets again, and it takes a little bit of time for the resetting. Okay, but remember our trigger zone. Let's just say this is the trigger zone here. Okay, and we get up to negative 55 millivolt. I only have one channel here, but you can imagine around this axon there are many channels in this stretch, many channels in this stretch, many channels. So this axon is, the membrane has many channels, many voltage gated sodium channels and voltage gated potassium channels. But at the trigger zone, the opening of this sodium channel, okay, we have a rush of sodium coming in. That's going to depolarize the membrane. It goes up to positive 30, so it should be able to depolarize the next channel in the lane, or in line, the next stretch of channels. Okay, but what keeps us going in from preventing us from going backwards? Okay, when this second channel is open. Okay, so this channel opens, let sodium rush in, then the inactivation gate comes in. That sodium that rushed in will cue the opening of this channel. And the opening of this channel will cue the next patch of channels down the line opening. And the opening of this channel will cue the next ones down. Okay, that is called the propagation. We're propagating so that we don't decrease the amplitude. We're re-upping all the way down the membrane. The other factor here is when this channel is open. This channel had opened before it, and now it got its inactivation gate. So when this channel is opening, the rush of sodium here would cause the value over here to be definitely over negative 55, but because this channel is inactive, we can't open it until this is reset. Okay, and remember this is happening very quickly. The rush of sodium comes in, boom, it's going to rush down here. This can cue this channel because this channel down here hasn't been open yet, so it doesn't have its inactivation gate. But when the sodium comes in here, cues this channel. When this channel is opening, this channel would be inactive. So that inactivation gate keeps us from going in a backwards direction. It keeps the action potential moving in one direction. Okay. And also the opening of one channel causes the one further down the line to open. This one opening causes this one to open. And so it's kind of like a positive feedback loop down the line to propagation of the action potential down in the one direction. Just to re-show the sodium voltage, this is the open state, it opens and then it is inactive. And then the channel, if we had channels, right, this is what the previous channel looks like when the other one is starting to open further down the line. Okay, so that's just saying when this channel is in this state, opening up, the channel here is in this state, right? And this channel here, when we're in the inactive state, we cannot open up the channel. So when this is opening, this channel can't be open, and so therefore we keep this action potential going down in that direction. Okay. So let's look at what we call refractory periods, and these are periods in which we have changes in which action potentials may or may not be able to take place. We have two. We have an absolute refractory period and a relative refractory period. Okay. In the absolute refractory period, no new stimulus can produce an action potential. Okay. So you can see here, it actually is probably more in this range between here and here would be your absolute refractory period. And what is causing this absolute refractory period? Okay, so say I have, we're recording at one stretch of the membrane, for the action potential, if you remember, our sodium, voltage gated sodium channels. When this channel is open, this one is closed. So or inactive, sorry, the inactivation gate is in. So in this portion of the membrane, I'm recording here, if these channels are in this inactivation range, I cannot get another action potential. So the absolute refractory period is caused by that inactivation gate. Until this channel resets, I can't open it again. And if I can't open this channel, I can't have my action potential. Okay, so we have this period here where we're inactive. Okay, once we're out, once these are reset, we are able to have an action potential. Okay, but that is the relative refractory period. Okay, in the relative, we can have an action potential. So say it's over here, those channels are reset, they can open again, and so I can get another uptick. The difference is what's happening in this period here. Where is my membrane potential? Yeah, I'm in that hyperpolarization phase. So let me draw it out here. That's a little, but here is when my inactivation gates come on. Between here and here, my inactivation gates are in place. Now once they've reset, I can have another action potential. But what do I need for another action potential? If I'm here, what's taking place? Can I have the same stimulus that brought me up to negative 55 before? No, because all I had to do was go from negative 70 to negative 55. But when I'm in this portion, my sodium channels are open, so they can come up again. But to get open, I have to bring my membrane potential all the way from here because I am in this hyperpolarization phase. Okay, so to get a second action potential in the relative refractory period, the stimulus must be greater. Okay, I have to make up that gap because originally I just went from the first one here, I went from resting membrane potential up to negative 55. But now, when my membrane potential, it is hyperpolarized at this phase, I have to go from here all the way up to here. So I have to bridge this gap here. Okay, so in absolute, the inactivation gates are in play. So I cannot open the channels, I cannot have an action potential. In relative refractory period, the channels have reset, but they are reset. My membrane potential is still in the hyperpolarization phase, and therefore I have to have a greater stimulus to make the second action potential take place. What else is important about the absolute refractory period? Right? Because when I'm in absolute refractory period here, down the line, this channel will be opening. So the absolute refractory period allows us to keep that membrane potential, that action potential going in one direction. Okay. Okay, so just kind of the actions that are taking place during the different periods. In absolute refractory period, the voltage gated sodium channels in that region are not reset, so I can't open. In the relative, the sodium channels now have reset, but I am still in the phase where my potassium channels are open. So to get up, I have to overcome the potassium channels. I must have a bigger stimulus to get that second action potential to take place. Okay. This is actually a good one. Again, I'd probably put the refractory period over here, but this is showing the state of the different channels during the absolute refractory period. Okay, where is the state of my channels? They haven't reset yet. Okay, and my relative, my channels have started to reset. Some of them, and not all of them have, but my potassium is still leaving. Okay, so here, channels haven't reset. I can't reopen them. Here, some of my channels have reset. I can reopen the sodium channels, but what's happening is my potassium channels are still open. Okay, so those are the reasoning for the refractory period. All right, so here's that slide where we're looking at if we measured all the way down the axon, you would see that the amplitude of the action potential is roughly the same all the way down the axon. Okay, it doesn't come down and decrease. Our graded potentials will lose steam as they move through the cell, but these stay the same, and that is because all along this axon we have sodium and voltage gated sodium channels and voltage gated potassium channels. So every stretch, or one channel opens the other channel, opens the other stretch of channels, okay, all the way down. So we're getting this rush of sodium all the way down because we're opening up voltage gated channels along the way. Okay, so any moment you will have the same channel because we have sodium stretch voltage gated sodium channels opening here, causes these to open, causes these to open. And that's the propagation effect, in that these channels, the previous channels, preceding channels, open up the next channels. So that's what you see happening as we're moving down. Okay, we have at the trigger zone, we got to reach that negative 55. Once we reach that negative 55, boom, there is an action potential. And remember, whether we reach negative 55 or positive 30, that is just cueing the action potential. The action potential is not going to be greater. It's going to look the same. But after the trigger zone, the cue to open up these rest of the channels is the opening of the preceding channel. So graded channels at the trigger zone open up the channels, but as we go down the stretch of axon, the opening of these channels will cause enough depolarization to open up the channels further down. And then when these channels open, it will cause these channels further down. So we have this re-upping every time. Okay. And that's called the propagation of the action potential. Okay, as you can see, here is the sodium channel. We open up. That cues this next channel, set of channels to open up. And remember our absolute refractory period. Here's a good picture of this taking place is when this patch is in open phase, the patch behind it is patch of channels is in the inactive phase. So when this channel is opening, this channel cannot be open again. The only channels that can be open are the ones that are further down the axon. All right, and then when this one opens, it's going to cue these to open. Okay, that is the propagation of the action potential. Okay, opening of previous channels or preceding channels causes the opening of the next channels down the line. So you can see every time we're getting rush of sodium in. So therefore, we're not losing any of the change in membrane potential. We're not losing any amplitude on it. Whereas with the graded potentials, we don't have this re-upping. We have a channel opening wherever in the cell membrane, and there are no channels to be further opened up. Okay, but in the axon, we have all these channels, and so the propagation, the word propagation, meaning the preceding channel causes the next channels to open. The opening of those channels causes the next. So we just propagate down the line. Okay, and then the absolute refractory period the inactivation gate. That's going to keep it from going in a reverse direction. Velocity. What would affect the velocity of this propagation of action potential? You can see there are several factors. One we're going to discuss is myelination. Okay, the oligodendrocytes and the Schwann cells, they produce that myelin on the axons, or those insulation wrapping. Okay. Diameter of the axon, and also temperature. Okay, but let's just say size and myelination are kind of the ones we'll focus on. So let's look at speed. Again, two key, diameter, and also myelination of the membrane. Okay. We get this myelination, we get this wrapping of these axons in here. So let's take a look. Okay. The myelination. Remember, these Schwann cells, they are making these little fatty insulations. Insulation is good because remember with our graded potentials we had leakage of current. But if we have this fatty insulation, there is no leakage. So if an action potential takes place, starts here, the current moving down here, there is none leaking out. So essentially all that current can be transferred to this next spot that is open called the nodes or Ranvier. Okay, so myelination will cause conduction to be faster in these axons. Okay, it's going to minimize the current leak. Okay, because remember we always have to be at least -55 to open the voltage gated channels. Okay, and so we have the myelination from the Schwann cells, and in between it is not completely myelinated all the way down. There are gaps in the myelin, and those gaps are the nodes of Ranvier. Okay, what do you think you might find in these nodes of Ranvier? We're on the axon, so yeah, voltage gated sodium channels and voltage gated potassium channels. Okay, just as you would see down the other axons. But in this case, we're only finding these channels in the unmyelinated region, in the nodes of Ranvier. Okay, you won't be finding sodium and voltage gated sodium and potassium channels in the axons underneath the myelin. They will only be found in the unmyelinated areas. So it is much faster conduction down in a myelinated axon due to what we call saltatory conduction. Right? That is the signaling, the electrical signaling, the action potential taking place down a myelinated axon. Okay? If you remember, in an unmyelinated axon, we have to have sodium and potassium channels all the way down. So every step of the way, we got to re-up, 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 re-up. But with saltatory, with the myelin sheath there, the depolarization that takes place here moves in this direction. And because we have this insulation, there is no leakage of the current. All right? So the current doesn't leak, so we don't lose the current, because remember, when we reach this patch, we still need to be at -55 millivolts. Okay, but now we don't have to open at every little stretch. We can open at the stretch, and because of this insulation, we can move the current here, and so it's basically the signal is jumping. This is much faster than every little step of the way we have to re-up. We can go boom, boom, boom. Okay, so this mode of conduction is much quicker than that of what we say continuous. Myelinated axons, there's myelinated axons and there's continuous ones. Continuous means they have channels all the way down the axon. Okay, but keep in mind that your cells, your neurons, don't make channels where the myelin is. We don't need the channels. Okay, they are only at the locations in the nodes of Ranvier where we find the channels, the voltage-gated channels. So this can cause problems though, having this myelination, because of demyelinating diseases. If you have multiple sclerosis, Guillain-Barré syndrome, okay, this is where the myelination basically gets broken down. Okay, and what happened? The myelin served to prevent conduction leak. Okay, because remember, we have to get to negative 55 millivolts to open up the channels. Okay, so what happens in these diseases is, you see, we get the rush of sodium in, we get our depolarization here, but there are no channels in this stretch of the membrane. That was once where the myelin existed. All right, if the myelin's not there to basically prevent the current leak, what's going to happen? We're going to lose current along the way, lose the signal, and when you reach this next stretch, this next node of Ranvier, there's a possibility we won't be at that negative 55 millivolts. All right, and what do we want to do? We want to send this signal, this electrical signal, all the way down the axon. Okay, we have to get it to the axon terminals. Okay, we'll see why we have to get the action potential and what the action potential cues at the terminals, but to get the signal down, we have to go all the way down the axon. And in these demyelination diseases, okay, you'll see what happens to the signal here. Can you signal through this neuron? No, if you lose the action potential, you're not going to be able to signal. Okay, and so you lose whatever this neuron's function is. Okay, many times vision, so this would only affect those neurons that are myelinated. Some of your highly myelinated axons are those that control your muscle. Okay, your muscles are these long axons, so they have to be myelinated, and so you get muscle weakness, difficulty walking, and loss. So many of the things that affect the muscle. Also, some of your vision is also myelinated. Okay, but this is a very serious disease. This would affect many, many functions of the body. It is not a pleasant disease. So, but the cause is that they have lost the myelin here and lost the function of it. Now current can leak, and that can disrupt the signaling of that axon. All right, so we've now seen that action potentials, once they're fired, they are of the same height. Okay, same amplitude. And this happens whether they reach negative 55, whether they reach positive 20. As long as it's triggered, it's triggered. Okay, it's going to go all or none, and it's going to go all the way. Okay, the difference, so how do we see a difference? Say your sensory receptors, they respond. We'll use touch for an example. When I just touch you lightly, you just feel a light touch, but then when you very firmly press into your skin, you feel a harder touch. And why is that? If the amplitude, if that sensory neuron, the amplitude is the same, how can your brain tell the difference? And how it does is that a larger stimulus will actually cause larger amounts or higher frequency of signaling. So this top one would be an example of, say, that lighter touch. Okay, you can see the frequency. Each of these lines here is a action potential, and you can see that neuron is firing kind of high frequency, but with this bigger stimulus, you can see the frequency of action potentials is much quicker. Okay, so that is how the brain is going to be able to interpret the differences. The frequency of action potentials is what codes for the stimulus strength, not the actual action potential, because these action potentials are of the same height. But you can see frequency-wise, the one with the stronger stimulus, right? Here's that strong stimulus compared to the smaller one. You can see the frequency of action potentials is what the difference is. Okay, so frequency codes for intensity. Okay, light touch here versus a strong pressure touch there. Alright, so we've done our graded potentials. We've looked at our graded potentials, those potentials taking place at the cell body and the dendrites. And if we have enough graded potentials to reach threshold, then we send that electrical action potential down the axon, that electrical signal. And remember, what's the function of the neuron? The function is to be able to talk and communicate with cells over long distances. So that action potential comes down the axon, and at the end, we reach the axon terminal. So now we're going to look at what takes place at the axon terminal to allow us to communicate to those cells, those target cells. Basically, the neuron, what the cells that the neuron's talking to are considered the target cells. So this depends on, we need signaling molecules. Okay, so it's actually going to be chemical signaling. We've done electrical signaling through the neuron, but when we get to the end and are talking to the target cells, we are going to be using chemicals, signal molecules. Right? And these signal molecules will be secreted at those axon terminals, and what we need is we have to have receptors at the target cells. So the neuron releases chemicals, and the target cell has receptors that will receive the chemicals and will get a change in that target cell, depending on what that signal molecule is. Now this takes place, we have the axon terminals coming down, and those would be considered part of the presynaptic cell, because when these terminals come down, there's the terminal, and here's, say, the membrane of the target cell. So this would be, the neuron would be the presynaptic cell. Before the synapse, this would be the synapse here. And this is where the chemicals are going to be released in. And then you have the target cell, the postsynaptic cell. Now the postsynaptic cell can be another neuron, but it can be many other different types of cells. Okay, it can be the heart muscles, it can be endocrine glands. Okay, it can be any type of cell, depending on what the cell the neuron is talking to. Okay, so we have these synapses, chemicals, and the ones we're talking about here, when we're talking about synapses, is these chemical synapses. We're going to spend the most amount of time here on chemical synapses. That's what we're going to talk to, but we are going to talk briefly about the electrical synapses. Okay, so chemical synapses is where chemicals are getting released. Okay. This is the most common type of signaling in the nervous system. Okay, is this chemical signaling. Okay, there is another type, electrical signaling, that's made up a little bit differently. It's not release of chemicals, and so we'll take a look at this first in these couple slides, but then the rest of the lecture will be focused on the chemical synapsing that is taking place here. So here we can see both types of synapses. We got both the chemical and electrical synapses. Here's the chemical, and where you can see the presynaptic neuron is releasing neurotransmitter, okay, the chemicals, into this synaptic cleft. And the target cell has receptors for that chemical. Right? This is the one we're going to talk to in great lengths. We're going to focus here just for the next couple slides on the electrical synapse. This is another setup, but we're not using chemicals. What we have are these connection proteins, these little gray guys here. And these connection proteins basically make up a gap junction, a channel between the first cell and the next cell. Okay, and what we can see, this is actually a connection between the two. It's like a tunnel between the two cells. So what we see here is if an action potential is coming down, that electrical signal can actually be brought across to the next cell. Okay, we can have, if movements are taking place here, those ions can also be moved into the next cell. Okay, we see that here. We're not going to see this a lot in the nervous system. This is few and far between for this. Most of the signaling, the majority, is chemical signaling. We will see this come up again, these gap junctions, when we talk cardiac, because this is part of the electrical signal of the heart is set up this way. Okay. But just looking closer, you can see the synapses. There's those connection proteins. These right here, you can see movement from the one cell, movement of ions can be brought over into the second cell. Advantages of this, we get this rapid conduction, and it also is going to be able to synchronize the activity of this cell with this cell, with the pre and the postsynaptic cell. Okay? And we'll see there are some in neurons and glia, but where we're going to see this again is going to be with the cardiac and a little bit with the smooth muscle. Okay. So the rest of the lecture we'll be talking not about the electrical synapses, but we'll be talking about the chemical synapses. Alright, so let's now get into the chemical signaling. We start with the chemical synapse. And that is that region here in the circle. You can see the pre-synaptic neuron. Its axon terminals come very close, not a physical connection, but come very close to the target cell that it is talking to. In this case, it is a neuron, but I've said this postsynaptic cell can be many different tissues. It can be muscle, can be endocrine glands. Okay, so there are many different tissues. It doesn't have to always be a neuron. In this case, the example, they are using a neuron. But the axon terminal is coming in, and it comes very close, and there is this synaptic cleft. Just a little narrow space between the presynaptic neuron and the postsynaptic cell. Here, the synaptic cleft. And that is where the chemicals are going to be getting released into. So looking closer at this chemical synapse, here is the presynaptic neuron. And what's taking place here is that action potential is coming down the axon. So what's taking place at the synapse is we're taking this electrical signal, this action potential coming down, and basically converting it into a chemical signal in the synaptic cleft here. And so the cue for these, you can see these little chemicals, these neurotransmitters, are sitting in vesicles. And if you remember our membrane transport, our exocytosis, this is where exocytosis is going to be taking place. And what is cueing the exocytosis of these neurotransmitters from the axon terminal into the synaptic cleft is the action potential. So the action potential is the cue to release these chemicals, these neurotransmitters, into the synaptic cleft. Okay, and then you can see the postsynaptic cell has receptors here that are going to bind to the chemical messenger, the neurotransmitter. So let's look at the steps that convert that action potential into the release of these signal molecules, to the release of these neurotransmitters. So that action potential is coming down the axon. Okay, we have a new player here. We have a voltage-gated calcium channel. All right, voltage-gated calcium channel. And if you remember, calcium is high outside compared to inside. So if that channel opens, calcium is going to be rushing in. Okay, so the Q, that action potential, change in voltage, right? It is going to cue those voltage-gated calcium channels that are located at the axon terminal, right here, to open. When those channels open, calcium rushes in. And now calcium is the cue or trigger for exocytosis of those vesicles. So calcium is actually going to activate these snare proteins, and we'll look at these in just a second on the next slide to get a better visual. But there are these snare proteins that result in the docking and the exocytosis of the neurotransmitter. Okay, again, we have action potential coming down. That action potential cues the opening of the voltage-gated calcium channels. The rush of calcium cues these snare proteins to result in docking of the vesicle and then exocytosis. Okay, so we're taking that electrical signal, bring that action potential coming in and cueing it for release of neurotransmitter into the synaptic cleft. Once the chemical is in the synaptic cleft, it can come down and bind to the receptor on the postsynaptic cell. So that is simply what's taking place. We're converting that electrical signal into this chemical signal in the synapse. So here's a better look at some of the, you can see they're almost like little tethers, these snare proteins. Okay, there's some that are located, this would be the plasma membrane here. And then you got these blue ones hanging from the vesicles. And that's the cue. The calcium cues the meaning of these snare proteins here. And now we get docking and we get release of neurotransmitters. So that's the calcium. The calcium cues this process here. Okay. Anybody heard of Botox? Okay, botulinum, it's actually a toxin, right? It smooths out the skin. What it doesn't do, what it prevents is your muscles from contracting. So if you inject it into your, say, your forehead where you have these lines where your muscles have been contracting, you can insert it or inject it in there, and it basically will prevent those muscles that are creating that line in your forehead from contracting. And how does it do this? It actually acts at these snare proteins. It will digest snare proteins in acetylcholine neurons. Okay, acetylcholine, we'll see later, is the chemical messenger to cue muscle contraction. And if you're not able to release, you break these snare proteins. In this case, acetylcholine would be the neurotransmitter in this vesicle. If you prevent them from docking and releasing, then you prevent those muscles in your forehead from contracting. Okay. All right, so let's look what happens to that postsynaptic cell, where the postsynaptic cell has the receptors that bind the neurotransmitter, those chemical signals that get released from the presynaptic neuron. I'm going to go in kind of the basics here of the signaling. We're going to go into a little more detail when we get into the cell signaling, but just so you have an understanding that you're getting a response in the postsynaptic cell. Couple of types of receptors we'll be looking at are ligand-gated ion channels and G-protein couple receptors. Okay. The ligand-gated ion channels is just simply that. Those are the chemically, you can think chemical-gated ion channels. In this case, the chemical is the neurotransmitter. We'll see later on, hormones also have receptors, and in that case, the chemical that's binding to the channels or the receptor is the hormone. Okay. In this case, chemically gated ion channels, a neurotransmitter gets released from the postsynaptic cell, and when it binds, you can see these channels are closed. Then you get binding here of the neurotransmitter, and that causes the opening of the channel. Now, if this was sodium, sodium would rush in and cause the cell to get depolarized. Okay, so these ion channels can change membrane potential of these postsynaptic cell. If this was a neuron, remember our graded potentials, with that might cause one of our graded potentials. If this was a sodium channel, we would have sodium rush in and we would get depolarization of this cell. Okay, so that's one effect that can happen at the cell. We can change membrane potential by opening up chemically gated ion channels. The most diverse and most abundant types of receptors, and they're going to have a lot of diverse functions, are these G-protein couple receptors. These are membranes receptors, okay, and binds a neurotransmitter. In this case, we can get multiple effects. What these G-protein couple receptors is, it has these G-proteins that are coupled to the receptor, and binding of this neurotransmitter will activate these proteins, the alpha, beta, gamma, these subunit proteins. Okay? And these activated proteins can then go and activate, sometimes can activate other proteins. Okay? We will see they can activate enzymes. They can also go and open or close channels. Okay, ion channels, so they can also affect membrane potential, but you will see they're also a diverse effect in that they can also activate many different other proteins, and we'll see that later in the cell signaling, and we'll see a little bit of it when we talk the monoamine neurotransmitters. Okay, but for now we got ligand-gated ion channels, and you can see the difference. This is a direct link between the ion channel. The actual neurotransmitter opens an ion channel, where here with the G protein, it is the actual activated. Okay, so this channel gets opened indirectly, not directly by the neurotransmitter. So a little bit different. This would be much faster. This would be a slower response. Again, slower, we're talking milliseconds to seconds. Okay, not super far behind, but we can also see that these G protein receptors can also activate many other proteins, and we'll talk about those a little bit more. Okay. All right, so the first neurotransmitter we're going to attack is acetylcholine. You see it abbreviated ACH. All right, these are, you may be familiar with this, you may have seen this in anatomy, they may have gone over this as the neurotransmitter you see at the neuromuscular junction, where the neurons meet up with the skeletal muscles. The neurons that secrete acetylcholine and the receptors that bind acetylcholine are termed cholinergic. So we have cholinergic receptors down here, and this would be a cholinergic neuron because it's releasing acetylcholine. And just like we've seen with release of neurotransmitter, okay, our neurotransmitter in this case is part of acetyl-CoA. The acetyl portion of acetyl-CoA is bound with colon, and there's an enzyme here that puts those two together to make acetylcholine. And the acetylcholine is packaged in a vesicle, and just with any neuron, it's waiting for that action potential. When that action potential comes, we get that cues the release of acetylcholine, and acetylcholine will bind to its receptor, and we will get an effect. So neurotransmitters, we don't want them constantly here. If this was at your muscle and the acetylcholine was just hanging out here for long periods of time, your muscles would stay contracted. So to get rid of the signal, there on this neurotransmitter, there is an enzyme, acetylcholinesterase. Okay, and you can see what it does. It cleaves acetylcholine back into the acetyl acetate group and the colon. And in this case, the colon will get what we call reuptake. It's brought back into the presynaptic neuron, and it will be put back in to make acetylcholine. Okay, so acetylcholine gets released, binds to its neurotransmitter, gets cleaved by acetylcholinesterase here on the postsynaptic membrane, and the colon gets reuptake back in. So one receptor, not written here, but is nicotinic. There are nicotinic cholinergic receptors. So one thing to keep in mind as we go through these is there are many types of receptors. There are multiple nicotinic receptors, and we're going to talk about another, the G protein coupled receptor for acetylcholine is a muscarinic. There are subtypes to these receptors. There's not just one nicotinic receptor. There's actually 7 to 8 subtypes, and you would have a different effect of acetylcholine binding to the different receptors. There would be a different response in the cell. So we can have these diverse effects in here because we could have differing receptors for that neurotransmitter. Okay, this could have nicotinic 1 or nicotinic 7, and you might get a different effect at the postsynaptic cell. But let's look at nicotinic receptors. Okay, these are the ligand gated ion channels, or chemically gated ion channels. Okay, the nicotinic or chemically gated ion channels. So when acetylcholine is released, let's walk through this. We have the action potential coming down. Okay, that action potential cues those calcium channels to open, and that calcium is involved in docking and fusing the membrane for exocytosis of the acetylcholine. Okay, the acetylcholine gets released in the synaptic cleft, and now the acetylcholine binds to the nicotinic receptor. Okay, it is a cholinergic receptor because it binds to acetylcholine. Nicotinic, meaning that it is a ligand or chemically gated ion channel. So what's going to happen in this case? In this case, you can see this allows sodium. So if sodium rushes in, what's going to happen to the cell? Yeah, we're going to get depolarization. Okay, depolarization. Of the cell. So in nicotinic, we can change the membrane potential by binding of the neurotransmitter to the receptor. We get a rush of sodium in. And then what else is here? We have the acetylcholinesterase. Okay, acetylcholinesterase gets broken down, breaks down acetylcholine, and then the colon is brought back and recycled. Okay, so that's the release at the neuromuscular junction, but again, nicotinic. Okay, we are also going to see there is another subset of acetylcholine or cholinergic receptors. Those are the muscarinic. Okay, in this case, the muscarinic, you can see are G protein couple receptors. Okay, and again, there is M1 through 5. So there are 5 different receptors, 5 different muscarinic receptors. And you can see we can get different effects depending on the muscarinic receptor that is there. In one muscarinic receptor, you can see binding of acetylcholine to the G protein couple receptor. It is a G protein, so that's going to activate the G proteins. But the activation of these proteins results in opening of a potassium channel that allows potassium out. And what's going to happen here is we're going to get hyperpolarization, right? If potassium is allowed to rush out, that's going to bring down, make the membrane potential more negative. Okay, in a different muscarinic receptor, okay, binding of acetylcholine again activates G proteins, but in this case, with this receptor, the G protein does the opposite. It closes potassium channels. Okay, and this allows for depolarization. Okay, if potassium is not allowed to leave, remember potassium's permeability allowed to leave makes it so it keeps it negative 70 millivolts. If we're retaining the potassium, that positive charge is building up and we're going to get depolarization. So you can see, depending on the receptor, even though the neurotransmitter is the same neurotransmitter, acetylcholine, we have 2 different effects at the postsynaptic cell, depending on which receptor is there. Okay, by having a different subtype receptor on the postsynaptic cell, we can get different effects in the postsynaptic cell. So we already discussed this, but the deactivation of acetylcholine, after it is released, if you remember, there is enzyme here, acetylcholinesterase, and acetylcholinesterase cleaves acetylcholine into acetate and choline, and then that choline gets reuptake back in and will be put back in to acetylcholine in the presynaptic neuron. Okay. This is important because we don't want this binding to be continuous. You don't want to sit there, your muscles completely rigid. You want them to be able to relax. So when we need to contract, we contract the muscle, but then the acetylcholine message is taken out and we can relax the muscle. Okay, so it is important that we are able to deactivate these neurotransmitters at the synapse. Another class of neurotransmitters is monoamine neurotransmitter. Okay, these are monoamine. Okay, they're derived from a single amino acid. In this case, for these, it is tyrosine. Okay, tyrosine is an amino acid. It is the starting block to make, you can see it is a multi-step pathway, but we get several neurotransmitters from. All right, there may be neurons that just create dopamine. All right, or another neurotransmitter is norepinephrine, and then we also, one you probably heard of, epinephrine, or also known as the old name adrenaline. Okay, but they are all created by tyrosine. We are going to see there are some others. Serotonin is going to be from tryptophan, and histamine is another neurotransmitter by histidine. Okay, but these are all monoamines because they are derived from a single amino acid. So we'll first look at serotonin. Okay. This one is formed from tryptophan, the amino acid tryptophan. It's heavy in turkey, and it has a soothing effect, a regulation of mood, appetite, right? And we can see the receptors for these monoamines, okay, are a G protein coupled receptor. Okay. They act through a second messenger. So in this case, we're not specifically opening an ion channel. What these G proteins are doing is activating an enzyme. All right, we will see this later, adenylate cyclase. This is an enzyme that converts ATP into cyclic AMP, which is called a second messenger. We're going to go in more detail when we look at the cell signaling in the next lecture. Okay, but this activation of second messenger, making the cyclic AMP, can activate many proteins and can also cause activation or opening of ion channels. Okay, but it can also have the effect of activating other proteins in the cell. But these monoamines their receptors are a G protein coupled receptor. And another one of the monoamines, the dopamine we saw, was derived from tyrosine. All right, that's single amino acid. And we have dopaminergic neurons. Okay, there are two major pathways to realize: the nigrostriatal, okay, and the mesolimbic. This one is one that is associated with a common neural disease you guys have probably heard of, Parkinson's disease. So this pathway is involved in movement. Okay, the nigrostriatal, the neurons of the nigrostriatal, the dopamine neurons, are known to be affected. Loss of these neurons in this pathway has known to be one of the signs of Parkinson's disease. Okay, the other pathway is the mesolimbic. Okay, and in here is the nucleus accumbens, is one of the regions of the brain that is stimulated with dopamine transmission. And this is the key to most addiction. All right, most addictive drugs, they will somehow influence or increase the signaling at the nucleus accumbens. It gives you a reward sensation. So even though some of those effects of those drugs are bad for you, you feel great and euphoric because it is activating this reward system in the brain. Okay, so the two pathways, one heavily involved in movement and the other an addiction or reward pathway. Okay. And then norepinephrine. Okay, that is further down the line, but it's a tyrosine. Okay, neurons can be first create dopamine, but then they have the enzyme to create dopamine or norepinephrine from dopamine, and they will have norepinephrine released as a neurotransmitter. Okay. In the CNS, we get, it's kind of the arousal. If you ever heard of narcolepsy, people that just spontaneously fall asleep, one of the drugs they use activates the norepinephrine system. So it gives them arousal. Okay. We will see sympathetic neurons. When we get to the autonomic nervous system, they are involved in signaling in the sympathetic nervous system. Okay, that's in the periphery of them. And we will see there are beta and alpha subtypes. We will see these when we get to that autonomic nervous system. Okay, but just like we saw with the serotonin, the monoamine signaling is again through G protein coupled receptors. Okay, again, we're going to get involved, a little more involved in the signaling of this, and so this will make a little more sense when we get there, when we get to the cell signaling. So many of your psychiatric medicines and so forth work on at the synapse. They work on either influencing, enhancing neurotransmitter transmission, or actually decreasing neurotransmission. Okay, so let's look at how this can operate. Okay, we're going to use a serotonergic neuron for this case. Okay, we can have serotonin gets released and binds to its neurotransmitter. Okay, what might be some ways to influence this? Okay, we can have what's called agonists. Okay, agonists are molecules that aren't serotonin, but when they can bind to the serotonin receptor and activate it just as if it was serotonin. So this, if we had an agonist drug, a serotonin agonist drug, we can bind it and we can increase the signaling of serotonin. We could also have an antagonist. What if we wanted to block the signaling? Okay. How could we do that? It's not shown here, but what if we had a molecule that could bind, kind of like a competitive inhibitor that we saw in the enzyme? It binds to the site in which the neurotransmitter was going to bind, but in this case, unlike the agonist, it does not cause an effect, and it also is blocking serotonin from binding. And therefore, it would decrease the signaling of the neurotransmitter. Okay. We can inhibit the neurotransmitter degradation. When we saw acetylcholine, how did we get rid of acetylcholine? There was that enzyme here that broke down acetylcholine. What if we blocked that enzyme? Okay, we blocked the degradation of acetylcholine. Then if this is acetylcholine receptor, we would have increased acetylcholine transmission. Okay, so, and what would happen? If we inhibited the breakdown of the neurotransmitter would stay in signal for longer periods, and therefore we would increase the transmission of serotonin signaling at the synapse. Okay, and another inhibition of reuptake. So reuptake in the serotonin neurons. Serotonin is released, and remember, we don't want the signal to stay here. We don't want a continuous signal. So how does serotonin and acetylcholine, we broke it down with the enzyme there. But for acetylcholine and norepinephrine, dopamine, they have these reuptake channels that will bring back the neurotransmitter. Okay, they will bring it back. What happens if we blocked? We inhibited the reuptake, so we blocked this. So if serotonin gets released, it doesn't get brought back into the presynaptic neuron. It will stay in the synapse for a longer period of time and will increase the signaling. So by inhibiting the reuptake, we increase the signaling. Okay. By inhibiting the degradation, like with acetylcholine, we increase the signaling. An antagonist, though, would decrease it, and an agonist would mimic the neurotransmitter, so it would also increase the signaling. Okay, so, and we have these different subtypes. What the drug companies are doing are basically finding drugs that are more and more specific instead of blocking multiple serotonin receptors, we can find them that would block just the 5-HT2 receptor. So that's kind of where the drug companies are starting to head, is to be more specific, because then they can resolve specific system and have less side effects. Okay, but you can see how these can affect neurotransmission at the synapse here. One example of a drug to increase signaling is a monoamine oxidase inhibitor. Right? You can see here in this dopamine, this is the monoamine oxidase. And basically what happens is when dopamine is brought back in, it is broken down by MA monoamine oxidase. So some drugs, some of the theories behind depression, and we also saw Parkinson's, would be a cause for low dopamine at the nigrostriatal pathway. Instead of preventing this breakdown, that would cause more neurotransmitter to be here. So when neurotransmitter is released, there would be an increase of dopamine released into the into the synapse. Okay, so that is one example. And we're basically inhibiting the degradation, inhibiting the breakdown of dopamine, so there is increased signaling at the synapse. You see some of your psychotropic drugs, ones like cocaine, their effects. Cocaine, known to be addictive, so you figure it's going to increase dopamine pathways, because those are the reward pathways, and dopamine does. It blocks reuptake of dopamine, norepinephrine, serotonin. Okay, so it blocks the reuptake, so dopamine signaling is increased in here. Okay, the signaling increases if this is neurons of the mesolimbic system. Okay, the nucleus accumbens, that's our reward center, and you would have increased reward sensation because you're blocking this dopamine reuptake. Some drug effects at the serotonin system, some of your classic hallucinogens give you hallucinations. Your LSD, your mescaline, your psilocybin, and mushrooms, they are serotonin agonists. So they act as agonists at some receptors in the serotonin pathway, and they actually, their effects are hallucinogenic. There are some additional effects to increase serotonin signaling. Your SSRIs, or the most common, or the first of these batch, was Prozac. Prozac's used for depression and anxiety treatment, and what it's going to do is increase serotonin signaling in the brain. And how to accomplish this is it blocks that reuptake of serotonin. So the serotonin neuron releases serotonin. Instead of being reuptake back in, it stays out here in the synapse and can increase the signaling there. All right, so we're on to the last two of the neurotransmitters we'll be covering, and those are the most, it's glutamate and GABA. Glutamate is the most prevalent excitatory neurotransmitter in the central nervous system, and GABA will be the most prevalent inhibitory neurotransmitter in the CNS. Okay. You can see this is widespread signaling, so you can imagine if we had drugs that affected these receptors, you would probably have a pretty profound effect. And one of those would be PCP. PCP acts as an antagonist to glutamate at specific receptors. Okay. This drug, PCP, basically almost mimics schizophrenia to as best of any drug can. It gives you hallucinations, but it also gives you the affective part. You just have no affect. So it basically gives you the effects of schizophrenia. Also, PCP, we're blocking the most excitatory neurotransmitter. These
people on PCP also don't feel pain. You'll see they get them in handcuffs, and basically these people on PCP will pop their shoulder out to try to get the handcuffs over, and they can do it because they don't feel any pain. Granted, they are causing damage, but they are not sensing that damage because they are blocking that glutamate signaling. Okay. And so glutamate is involved in what we call synaptic plasticity. Your neurons, you don't gain neurons. As you learn, you're not gaining more neurons. What you're gaining is you're enhancing the activity of signaling at certain neurons. You're enhancing the signaling here. Okay, if we get more extensions or if we increase receptors here, we can enhance the signaling. Okay, that is a synaptic plasticity. Okay, it enhances. We can potentiate, enhance activity at the synapses, or we can inhibit. You can depress and decrease the synaptic activity. And this is kind of what you guys hopefully are doing now, is as you're learning, you're doing facilitation. Okay. That's where it comes into play. Like if you repeat, you're starting to facilitate, and you're basically enhancing the synaptic activity on, say, hey, I need to remember synaptic activity. Okay, it's through potentiation. If you repeat this, you're enhancing the activities at those neurons, and when it comes test time, you're able to recall this stuff because you have this enhanced activity. Okay, so we're going to look first at long-term potentiation, and then we'll look at depression. In long-term potentiation, that is handled through glutamate signaling. Okay, you're going to increase sustained changes in the quality and quantity of connections at the synapse. Okay, again, we don't increase neurons, we can increase synaptic activity. Okay, and these are related in learning and memory. So let's take a look at what's taking place to increase this or enhance the synaptic activity. Okay. It involves two types of glutamate receptors. We have the AMPA receptor. We're going to see that is going to cause depolarization in the postsynaptic cell. Also located is an NMD receptor. The NMD receptor is unique in that it's blocked. It has a molecule of magnesium, okay, blocking this channel. When this channel is fully open, it allows calcium as well as some sodium and potassium to flow through. Okay, and we're going to see when open, the receptor is bound to glutamate, but it also has to be depolarized. Okay, and we'll see why this is the case, why we have to have both binding of the receptor to glutamate and also have the cell depolarized. But when we have this, when this all takes place, we're able to increase the quality and quantity of the synaptic connections, and we'll see what takes place to make this be increased. Okay. And we're going to see that postsynaptic cell becomes more sensitive to glutamate. There's going to be an insertion of more glutamate receptors on the membrane. If there is more receptors, that postsynaptic cell can respond more strongly from glutamate being released. Okay. The postsynaptic cell will also release paracrine that will cause increased release of glutamate. So we have twofold effect. We have more glutamate being released by the presynaptic neuron, and we have more receptors on the postsynaptic cells, so we can get a stronger signal. So as you learning, and you keep repeating, you keep repeating this process and opening these channels, you're going to increase more receptors and you're going to increase more signaling from the presynaptic cell. So the more you do this, the stronger the signal becomes, the more these receptors are increased, and the more neurotransmitter gets released. And when it comes test time, if you've done enough of this, you'll have the memory there to recall it. So let's take a look. Long-term potentiation. Here's that presynaptic axon. Okay, here is the AMPA receptor, and here's the NMDA. We'll see this magnesium is actually lodged in here. Okay, what happens first? We get release of glutamate. Okay, glutamate's going to go down and bind to the AMPA receptor. The AMPA receptor is a sodium channel, ligand-gated sodium channel, so it's closed until glutamate binds, and now sodium can rush in. That's going to depolarize the cell. Okay, if there is enough depolarization and this NMD receptor is bound by glutamate, and there is enough depolarization, the depolarization is going to eject this magnesium. So that's why we have to have both binding to open the channel, and we have to have depolarization to eject the magnesium. But once that takes place, calcium can rush in. Okay, calcium is going to activate second messenger pathways, and what that's going to cause is for more of these receptors to be inserted into the membrane. Also, that paracrine signaling, this postsynaptic cell is going to send chemical messenger over to cause this neuron to release more neurotransmitter. So by this taking place, these actions of the AMPA and the NMDA, we get increased release of neurotransmitter, and now we have more receptors here, and so we're going to have even more increased depolarization because we have more AMPA receptors here. So we're going to strengthen this signal. All right, so as you learn, all right, I'm learning about synaptic plasticity. Boom, fires, synaptic plasticity, glutamate, boom. We're making these connections and making them stronger, so when it comes test time, you're able to remember, because this signaling will be very strong. All right, on the opposite end of the spectrum, we have the most prevalent inhibitory neurotransmitter, and that's GABA. And GABA is basically they are gated chloride channels. Remember, chloride is negative, but chloride is high outside and low inside. So by it moving in, it will inhibit the cell. It will bring the membrane potential down. Okay, so it will cause inhibition. So GABA binding here opens the channel, and now chloride can rush in. Okay, causes the cell to hyperpolarize. Right? This is kind of like, remember we saw those excitatory postsynaptic where we had the depolarization. This here would cause the inhibitory postsynaptic cell because this would inhibit the cell. Okay, so they are chemically gated chloride channels, so they're going to cause inhibition. Some of your common drugs, your benzodiazepine, your Valium, okay, many of your sleep medicines, Sonata or so forth, they act at a specific one, or there's GABA A and GABA B receptors. It acts at one or both of those receptors, okay, as an agonist, because it increases inhibition and increases kind of the sleepiness of the brain. Okay. So that is it for nervous system. I know it's a long haul, but we'll have a short lecture on
neural signaling or cell signaling, and that will kind of bring in what's also happening here at the receptors.